good afternoon or good morning to everyone. I hope you all see my screen and the cursor on your screen. I just would like to warn you that I have some people in my room, so I might speak louder than you would expect. So I will start and go on the presentation now. So today, the aim of this presentation is to show you how Oncodesign can help you to support your drug discovery project. At every step of your preclinical development, I will try to emphasize some in vitro and in vivo assays we have developed in-house to routinely assess your compounds, whatever they are. And of course, I will also explain you what are our capacity through example of customized assays. Well, because you registered to this webinar, I'm sure you're well aware that immunotherapy is a hot topic right now in research and development and in clinical trials. Usually you have different approaches to develop immunotherapies, either using cancer vaccines or using cellular immunotherapy or developing either small molecules or biologics, antibodies, bispecific and so on, that will target either immune checkpoint inhibitors, immune checkpoint stimulators, or a combination of both, and so you have lots of different combinations ongoing. So because of the importance of this field growing up in the medical field, there are a lot of new companies and new projects that are being developed right now, and actually everyone is facing some issues in regard of the tests they can apply to efficiently support their drug discovery program, because dealing with human material is not always easy, and it's not easy to provide reproducible results. So whatever is your program, you usually have to go through different phases, which are summarized on this slide. So phase one is the target validation, in which, well, you find your target of interest that might influence the prognosis of the patient. Then you enter a hit selection phase, in which you have a screening of a bank of molecules to find a hit. And once this one is identified, then you have quite a long-term and robust effort in different phases to bring your hits into the candidate selections. So you have to put in place a robust and efficient flowchart, and that's where we can help you. So now Oncodesign have all the capacities to support your efforts in that aspect, either on a standalone basis, but now on a more integrated basis. Speaking of flowchart, I just had a couple of slides just to summarize a classical flowchart. So I took one that is usually applied when you develop small molecules, but you could easily adapt it when you are developing biologics. So usually you start by a batch of molecular synthesis by chemists, and this chemist provides the molecules that have to go through a first batch of assays. Usually you take assays to assess the potency of your compound, either biochemically speaking or in cellular assay. Or you also have tests to assess the physical properties, and you start to test the developability of your compound by, for example, microsomal stability assays. Once you have compounds that go through this this batch of assays, then you have a second batch of assays to set up. That includes phenotypic assays that is more related to the indication you want to address. And then you have also DMPK assay, the pharmacokinetic and developability assays, such as raw dose ranging and drug-drug interaction to follow up. Once you have molecules that go through these different phases, then you can include the third stage, which includes whole blood assay and pharmacodynamic and pharmacodynamic tests that will help you to predict the dose that will be used for the last stage of your program, which is the preclinical assay, in which you want to assess the efficacy of a compound in vivo. And of course, you have to start to think about the toxicity and the safety of your profile to anticipate the regulatory phase. So just as a reminder here now at Oncodesign we have, well, it should pop up in green, but it looks like yellow on my screen. But now we can help you in all these different phases. We have medicinal chemists and chemical abilities. We can perform a biochemical assay, and we have a huge now DMPK department that can help you to support your project. But for the purpose of this presentation, I will focus on the blue boxes, which represent the cellular assay and the preclinical assay. Krista, the floor is yours for the poll.
Yes, okay, thank you, Jocelyn. So I open the second question, which is, do you have any forthcoming project related to the current presentation? And I'll leave it open for about a minute. We're looking forward to your answers. All right,
Okay,
Jocelyn, you can, if you want.
Okay, I keep going. So having that said, so I just repeat this slide because this will be the thread of the rest of the presentation. It represents all the stage in which we help you to support to your drug discovery efforts. So I will start to, logistically speaking, where we start is the screening phase. So in that regard, at Oncodesign, we have developed a high throughput screening assay regarding the immunogenic cell death assays. As you probably know, some chemotherapeutics can induce a type of cell stress and death that is immunogenic, that converts the cancer cells into a vaccine that stimulates the immune response. Luckily, very clear readouts have been defined. They are called the DAMP and includes creatinine, HMGB1 and ATP. And those DAMPs are released during the ICD by the tumor cells. And this one recruit and can activate the immune cells, such as the dendritic cells, monocytes and T cells. So we have developed a high throughput screening with every one of these different readouts for the ATP, HMGB1, and the calreticulin, in which we make sure that we are subtoxic dose, and we see an increase of the signal. Here you can see on different cell lines U2OS, MDA-MB231, and HEPA-16. The increase of ATP level at non-toxic doses of docetaxel, for example. And you can observe the same trend regarding the HMGB1 and the calreticulin. So this kind of assay have been designed and is now in production in our Paris facility, and we can make campaign of screening to up to 20,000 compounds. We are right now trying to go further by developing a phenotypic assay ICD. This assay is under validation, so the principle is simple. You take some spleen from different mice, you isolate from them dendritic cells, and you put in plates, you co-culture them with tumor cells. In this slide it's CD26 that were previously loaded with DIO, which is a cell tracer. After 1 day of incubation in the presence of your treatment, you follow by flow cytometry CD11C, which is a marker of DC, and you follow the DIO fluorescence as well. And if you look at a double positive population, it means that you have disease that have performed phagocytosis on the tumor cells, and as a consequence, they incorporated the DIO. Okay. As a proof of principle, we used the chemotherapeutic dinaciclib in this assay. So you have here the results in one experiment. Here in the co-culture treated with the vehicle, so you still have a small amount of double positive cells. Then when you add a very small concentration of dinaciclib, which has been shown in ICD on the high throughput screening to be non-efficient. You don't see any differences. And when you have the correct concentration for ICD, then you can observe an increase of the double positive fraction, meaning that you increased the activation of disease in response to ICD induction. We also have developed lots of different phenotypic assay in immuno-oncology. It's not at the same level of throughput, but these phenotypic assays have been developed in-house and are now usable. These assays include different human population, PBMCs, T-cells, prenocytes, dendritic cells or macrophages, and we can work from human tissue, human blood or murine material. And we have different readouts, cytokine secretion, by flow cytometry, ELISpot, and so on. And I will go on after a few examples in more detail. I just would like to pinpoint that what is very, we are quite lucky in France that we have a good access to fresh blood, and in that regard, this is a plus for us because all of these tests have been developed to be tested on at least 3 different donors, just to include some donor-to-donor variation. So I will go now through some of these tests, but really quickly. One of those tests is a PBMC activation assay. It's a 96 well plate based assay, so we can use it either for screening phase of a very small bank of molecule, and of course it can be used as a support of your flowchart during the hit to lead to the candidate selection phase. So this test starts from whole blood from healthy donor. We isolate the PBMC using FICAL, and then we see the cells in 96 well plates. Then we perform on-auto priming, and we add some compound up to 10 doses per compound, and then we follow up the secretion of IL-2 by alpha-LISA in response to this stimulation. So we set up 2 different kind of assay, starting with PBMC. One we called screening of naive PBMC. So it's pretty simple. You take PBMC, put a stimulant, and you follow up IL-2 secretion. And we just want to make sure that this is robust along donors, so we have a good stimulation every time. Or you can use this primed PBMC assay, which is a 2-step process. In the first one, you stimulate, you titrate the PBMC with CD3, CD28 to estimate the priming concentration, which is roughly the EC50. And then you use this concentration to stimulate the PBMC and look at for top-up effects of your compound of interest at different doses. Regarding this test is a Tcell mediated cytotoxic assay, and we use as well 96 well plate. So again, we can use it for a campaign of small bank of molecules, or we can use it as a support, a routine support for your program. So in this test we use whole blood, we purify the T cells. We can either use them fresh or after freezing them, and we see them in 96 well plates plus or minus compound. We incubate the plates for a couple of days, 3 days, and then we co-incubate them with a Regi tumor cell line, which was previously loaded with calcine, which is a fluorescent dye. And after 3 days of incubation, we follow by flow cytometry the population of calcine positive cells in response to the stimulation of T cells. So if you have an activation of your T cells, it means that you will kill your Regi cells, which means that you should observe a decrease of the Regi population dose dependently of your compound. So this was exemplified here with an assay that was performed on 2 different donors in parallel, and you can see on the y-axis the signal related to the vehicle, and you see that along the concentration of, here it was CD3 that was used to stimulate the T cells, you see a dose-dependent decrease of the signal in response to the stimulation. Just for your information, right now we are starting to develop and validate this assay, but we changed the readout because we wanted to see either the Regicell but also the Tcell population. So we are now validating a test using CD19 to follow the Regicells and the CD25 antibodies to follow the T cells and their activation status. So you have a double readout in this assay. The last example of this ready-to-use assay is the M2 polarization assay. This is the low throughput assay so far because it's done on the 12 well plates, so we can use it to test a small batch of compound every time. And we start from the whole blood. We isolate fresh PBMC. We purify the CD14 plus cells, which are the monocytes. We seed them, and after differentiation, we polarize them in the presence of the compound of interest at different concentration, and we perform a phenotyping assay by flow cytometry using different markers. The most interesting ones are represented here. It's the CD206 and the CD209, and you can see that when you stimulate the M2 polarization, you have an increase of these two different markers as compared to the M1 polarization and no polarization at all. So we use this assay to test the IPI549 compound, which is a macrophage M2 inhibitor that is currently in clinical trial. And we tested it at 3 different doses, and you can see the result here. It's roughly that on this CD206 marker, we see a decrease either of the percentage of positive cells or the MFI, meaning that we have a decrease of the M2 phenotype in this assay, which indicates an effect on this compound on M2 polarization. For your right now, so this was the 12 plates, and now we are trying to miniaturize the assay to be able to assess more compound and more concentrations per compound. So this was a picture of the assets we have in place, but we also have a broad experience in developing and implementing customized assays. I just wanted to give you some examples. So there is one in this slide and a couple on the next slide. In these slides, this is an assay we developed for a drug discovery program to support it from the hit identification up to the candidate. This was a test performed on a weekly basis, and we tested more than 300 compounds. So we first had to set up the test. So this test was set up by using whole blood, extracting the T cells, seed them in 96 well plates, stimulate them with CD3, CD28, and then treat them with different concentration of compound. And the readout was the phosphorylation status of the target protein that was quantified by alpha ELISA. So this test was set up. We had to show the reproducibility, so it was okay, and then we had to set up a quality control follow-up. The bottom part of the slide shows representative results of assessing different compounds on this assay. The histogram shows that we almost always had a good stimulation by CD3, CD28, whatever the donors. And then the three next graphs show the dose response of compound. Here is the reference compound we use for quality control to just check that we didn't have variability between the different experiments. And then two results from compound A and compound B, for which we could calculate the IC50 and then communicate this IC50 to the project team so they could include the results in their analysis for the drug discovery program. We also have a strong experience in other assays such as ADCC, ADCP, or either immunohistochemistry assay in a very broad range of human cells. For example, we Here is represented results on an ADCP assay that was performed on HT29 colon tumor cell lines that was co-cultured with purified neutrophils. We also set up an assay in which we differentiated PBMC in gamma 9 delta 2 T cells in order to assess the interferon gamma secretion. And finally, the last example I wanted to emphasize is this kind of ADCP assay in which we took whole blood, we purified PBMC monocytes, we differentiated them into mature dendritic cells. These ones were vaccinated with a vaccine a client gave us. Then these vaccinated DCs were co-cultured with T cells for a week, and then co-cultured with tumor cells that were previously loaded with calcine. And at the end of all of this process, what we wanted to see is the percentage of lysis, which was determined by flow cytometry as a decrease of the calcine positive cells in presence of different ratio of DC. So when you increase the ratio of DC, meaning that you have more DC, you have a better effect of the vaccine. And I think, Krista, now you have a second poll to present.
Exact. Yes. Thanks, Jocelyn. So our next and last question is the following. What makes you choose Oncodesign among other CRO? And we'll be happy to have your feedback on this. I'll leave it open for a minute, and Jocelyn, you can keep going on the presentation.
Thank you, Krista. So now we have talked about in vitro and ex vivo assays, but of course a critical step in every preclinical project is a choice of a relevant animal model to assess the efficiency of the compound. Once you have, after a different iteration of chemistry and antibody production, selected potent and efficient compounds displaying a good pharmacokinetic profile. So usually in immuno-oncology model, and this is very specific for syngeneic model, usually you have to deal with a balance between three different parts. The therapeutic you want to use, either it's an antibody, a small molecule, a vaccine, or a T-cell. The preclinical model you want to use, because it depends on the indication of interest and the presence of the human population of interest within this model. And of course, the human population you want to assess and follow up during the process. So at Oncodesign, we have a broad experience in immunology models. I don't have a precise number, but I think we have dealt with hundreds and hundreds of different projects amongst the 10 to 15 last years. So as just to represent where we are, so we're in the preclinical model. So you have on the left part, the right part of the slide, a table summarizing the different syngeneic model we have, and in which we have tested standard of care in a checkpoint inhibitor or checkpoint stimulator with their representative efficacy, partial efficacy, or lack of efficacy. So as you can see, we have quite an impressive number of validated models. A second part of every preclinical model is how to assess the immune population. So internally, we have quite a good experience to do that by flow cytometry. We just have developed an in-house platform, and we just have new standardized model, and we developed 4 on syngeneic model, one for T, NK, and B cell, another panel for T cells more dedicated to checkpoint inhibitors, another panel dedicated for MDSCs and dendritic cells, and the last one for tumor-associated macrophages. So we have now a vast experience in using these panels in all kind of tissues, tumor tissues, spleen tissue, lymph node, blood, and so on. Of course, we can also propose services for immunohistochemistry if you are more focused on question that matters to localization of your markers. This slide is represented a case study of one model. So in this slide, it's the CD26 syngeneic model that was treated with CTLA-4 antibody. You can easily here see on the left part of the slide that when you follow up the tumor volume, you can see that when you have a huge difference between the control treated group and the CTLA-4 treated group with a decrease of the tumor growth. And by flow cytometry, by following the T-cell population within the tumor, we could observe also that when you treat the mice with CTLA-4, you increase the CD8 plus on-Treg ratio, meaning that you increase the population of cytotoxic T cells, and you decrease the immunosuppressive Treg population. So this is typically the kind of result we have when we use our syngeneic model. We also can propose humanized models, so those are more customized, and we can use for those ones either tumor cell lines, for human tumor cell lines, or patient-derived xenografts. Well, the challenge to find a good balance between the therapeutic preclinical model and the human population remains the same. And well, except that you have a broader choice of model available, because since we do tumor models, we do that for since more than 20 years, so we have developed more than 400 tumor models, and now we have more than 200 characterized PDX models. Regarding the human population you can use to humanize the mice, we have experience with PBMCs, with immunization using hematopoietic stem cells, T cells, by immunizing the mice with gamma 9 delta 2 T cells that were differentiated from PBMCs. We also had projects dealing with autologous T cell and dendritic cells immunization from monocytes. And actually, we are now in a phase where we start to test the sensibility of T cells from the compound prior to humanization in order to find which donor is the most responsive and select this one to humanize the mice. Once those mice are humanized and the tumor model is picked, we have a number of readouts. Of course, we have the basic ones with tumor volume and MRI and so on, but you can also have flow cytometry, immunohistochemistry, and well, we have a vast experience in Luminex and ELISpot as well. So I told you at the beginning that this was more customized, but we still have performed more than 150 different projects using this kind of model. So we start to have a good experience with that. I picked just a case study just to show how we would do for humanization. So in that case, I took a project in which we humanized the mice with autologous T cells and dendritic cells plus a tumor cell line. So at the beginning, we took whole blood from different donors. We isolated the PBMCs, and we had the double selection, a negative one to select the T cells and a positive one to select the monocytes. We obviously double checked that the yield of purity was good by flow cytometry, and then in the next step we differentiated the monocyte into immature dendritic cells, and at the end of this process, which took 7 days, we checked that we lost the CD14 signal and increased the CD1C signal, meaning that we had dendritic cells, and that the maturation marker CD83 and CD86 were not increased, meaning that we indeed had some immature dendritic cells. And finally, we mixed these T cells and dendritic cells from the same donors with tumor cells, and we injected them subcutaneously to different mice at different ratio, and we follow up the tumor volume. And on the graph is represented in black lines the tumor volume of mice that were not humanized, and you can see a kind of decrease of the tumor growth with the highest ratio of disease that was used. And in parallel, we tested the compound of the client, but this is unfortunately not something I can disclose in this presentation. So really, if I have a take-home message to give is that we are now working on immuno-oncology since 10 to 15 years, and we have developed a good batch of reproducible and robust vitro and vivo models that could really help you to support your drug discovery project. We are also always keen to develop new kind of assay, whether vitro or vivo, to support your project. Our goal is to integrate these assays into your flowchart to support you on a regular basis. So you just have one outsource that will support this part of your project. And as I quickly mentioned in the previous slides, because we acquired new facilities and we also incorporated new expertises, now we can provide what we call an integrated drug discovery service that would include, besides the biological part I showed you during this presentation, the medical chemistry part, the DMPK part, and the bioanalytical part. So that's it for my presentation, and I'm looking forward to having your question.
Thank you, Jocelyn, for your presentation. It was great. We are now ready to answer your questions if you have any. So we have not yet received any question, but feel free to use the chat to ask if you have any. We will be happy to stay online for a couple of minutes to answer them. Otherwise, if you have any further question coming up in the future, do not hesitate to send us an email, and we'll be more than happy to answer them. The presentation will be available for replay within about an hour, and you are also able to download the presentation using the WebIKEO Oncodesign channel. So we are looking for your questions, and we thank you for attending the presentation today. Jocelyn, looks like there is no questions coming up right away. So looks like you were
That's good for
very
me.
clear. That was a very clear presentation.
Thank you.
We are more than happy to answer any that will come afterwards. Of course, you have the contact details showing on your screen, so do not hesitate to send an email, and we'll get back to you by email.
We just have a question.
We have one question coming up. Okay, so it was good to stay on the line. So the question, Jocelyn, is the following. Phenotyping assay for Tcell mediated cytotoxicity assays. Is there an impact on using frozen T-cells versus fresh T-cells? And what would you recommend?
Okay. Well, usually you have to test both, because what we have done for the or you are looking for T-cell mediated. This so far we have tested T-cells that were frozen, but we have tested in different other assays and during the validations the difference between fresh and frozen ones, and we couldn't see any difference. But having that said, I think this is something you have to test for every time you set up a new assay, just to make sure that using thawed T-cells won't impact their ability to proliferate and differentiate and to be activated.
Okay, thank you, Jocelyn.
You're welcome.
Any more questions? We are still on the line, and happy to answer any questions coming up. looks like we don't have any further question asking us directly online. So I wish everyone a good afternoon in Europe, a great day in the US, and we are looking forward to hearing from you. Thank you very much for attending, and thank you, Jocelyn, for the presentation.
Yes, thank you everyone for your attention. Thank you.